bipolar cells, when they go into the light, we get less of the positive charges into the cell. That's what causes the hyperpolarization, right? Now, the same thing happens with our on-bipolar cells in terms of the photoreceptor level. When we go from dark to light, the photoreceptor still hyperpolarizes, but an on-bipolar cell will actually depolarize in response to that light or that hyperpolarization from the photoreceptor. That's because if we look at what glutamate is doing in the membrane of these bipolar cells, is actually normally inhibiting a channel that allows positive charges into the cell. So when that glutamate is no longer present, this channel the inhibition is relieved and the positive charges rush into the cell. That allows this cell to depolarize in response to the light. Yes?
So then if we went from light to darks, then that would match the membrane potential of the on-bipolar cell instead of going from dark to light.
Repeat that again. You're saying if we go from light to dark, here we would go from hyperpolarized to depolarized.
Yeah, okay. So it would match the on-bipolar cell. The membrane potential. I'm only referring to the membrane potential. Like the curve.
But that would change what's going on here, right? Because if this was instead depolarized, we would get more glutamate being released. So it would turn this into this situation where the glutamate there would inhibit basically this channel, preventing the positive charges. So both of these graphs would be inverted if we were going light to dark. That's a good question. All right, if there are legit questions, I'm willing to keep doing review instead of going on what we're supposed to talk about today. What? Well, you gotta ask a good question then. Any takers?
It might be a little more general, but I was a little confused when you were talking about mechanosensitive ion channels. Can you explain that again?
Sure. Do you mean in like somatosensation?
Yes.
Yes. All right. So in general, the idea with the mechanosensitive ion channel is that it's not like responding to a chemical. It's not, it's not responding to light, I guess you could say. It's actually responding to some sort of mechanical stimulus. And so we haven't looked at exactly how that mechanism works, but maybe it's stretched. As the membrane is stretched, the channel, that's a mechanical stimulus on the channel. So that allows the ions that would likely open a channel as it's being stretched or manipulated. Same thing with maybe pressure, right? There's pressure on the membrane that could lead to a mechanical stimulus that opens or closes an ion channel. Those are probably the main things. Yeah, and so if we go to so we talked about how those channels mainly stretch, or by proteins that are connected to, if there's force that's kind of pulling it one way or another, that's what actually opens the channel. Tiffany, did you have a question?
Yeah. So the ganglia receptor of the eye.
Okay.
Can you explain a little more about different firing action potentials on the on and off center? It feels like the circle and the target.
Sure. Okay, so we just did photoreceptors and bipolar cells. All right. When, we can kind of think of it as a one-to-one. In reality, it's not quite a one-to-one situation, right? We have kind of situations like this, where there's a few photoreceptors that are all kind of synapsing onto a bipolar cell. And we start to gain this idea of receptive fields, right? And because of the kind of identity of the photoreceptors that are here, and the characteristics that are going on in them. Also the fact that these center ones are directly synapsing onto our bipolar cell. These surround ones are still contacting that bipolar cell, but through the input of horizontal cells. So the setup allows them to behave a little bit differently. All right, and so we're still looking at the bipolar cell level, but it kind of applies just the same to the ganglion cell level, is that there's sort of this center that this bipolar cell is really sensitive to. And depending on whether this is an on or off bipolar cell, in this diagram I think it's an on-center bipolar cell. On bipolar cells depolarize in response to light. So when the light shines right onto the center of that receptive field, that's what we get. Okay? Now, light in the surround is going to be a little bit different, and that's because of these photoreceptors and how they actually interact with the bipolar cell. And so in this case, we have an on-center bipolar cell, meaning that it's going to depolarize in response to the light. What do we have in the center here, right? Dark, no light. So it's going to hyperpolarize. In this specific instance, right, this is just kind of hypothetical, right? Would we ever have the situation where light was only surrounding and not in the center? Probably not, but you know, we might have things if there's gradations of light over this receptive field. In this case, right, these ones are kind of going to inhibit the input that's here. It's already hyperpolarized, and so that just kind of further incentivizes the fact that this bipolar cell is going to be really hyperpolarized and not really sending signal to the next level. Yes?
For dark current in the memory potential of broad photoreceptors, would there be an influx of calcium and sodium into or through the channel? Or is it
yes.
not associated?
So, let's see. We talked about that a little bit more with adaptation.
Mm-hmm.
So, if we look at of course I can't find it.
I guess this question also follows along with it. As cyclic GMP binds to the sodium channel, right, in the dark, calcium has to, like, some calcium has to go in, right?
As cyclic GMP binds to the channel in the dark, yes.
Yeah, so it's both calcium and sodium?
It is.
Is that the same thing that occurs during, or for the dark current too? That causes the hyperpolarization?
Correct.
Okay.
Yes. There's, right, we also said, I kind of left this out at first, and then when we talked about this idea of our photoreceptors adapting, that there is this channel that the cyclic GMP binds to allows sodium and calcium through. Okay? The sodium is kind of what we said, okay, this causes depolarization, yada, yada, yada. The calcium actually has like a little bit of a different role in that it inhibits the enzyme that makes our second messenger. Okay? And so there's this kind of constant like feedback pathway where, okay, let's say we make a lot of second messenger because we're in the dark and the GPCR pathway, light pathway, is not on. The cyclic GMP binds to here. That allows everything in. All right, and then we have the depolarization in the cell. But we said, remember how, like, okay, you're in a dark movie theater and you walk out and it's like blinding light. But over time, right, at first it even like hurts your eyes, or like, you know, same thing. Your roommate turns your light on in the middle of the night. But over time, it's like we're not feeling that same, you know, pain or intensity in our eyes, even though the light level hasn't changed. And so it has to do with this pathway that as this channel opens, it's also allowing calcium in. Calcium inhibits the formation of the second messenger. So it's going to kind of slow down the binding here. This channel is going to close. Now, it's not like it's going to just permanently close because depending on what stimulus it is, in this case, light, right, I guess light would be degrading that, but it basically allows it to be able to adapt no matter what the situation is.
So there's not a steady flow of calcium. There's just a steady flow of sodium.
Right, it's kind of a play. Like if we're in the dark, we've got lots of this around, it binds here, it lets everything in. Okay, but if that was the end of the story, we would just have kind of this constant, depolarization in that cell. But things like also when you're in the dark, your eyes start to adjust a little bit, right? And part of that is because the calcium comes in and inhibits this, right? If we're in the dark, this is present, but as the calcium flows through, which happens in the dark, it inhibits the formation of more of this. And so that allows you your photoreceptors to kind of be primed and ready for the next stimulus to come along. All right, and as this inhibits this, we're going to decrease this, but we're still in the dark. So the G protein pathway is still being activated not activated, which means we have lots of this around. It's still being made. Yes?
How does that transfer, become, what you see, that adjustment in your...
Well, that's right. Everything happening in the photoreceptor level are just hyperpolarizations and depolarizations. So, right, and those cells release glutamate. So depending on how depolarized means how much glutamate is released. That glutamate sends information to bipolar cells. Those bipolar cells send information to ganglion cells, and those axons travel way up to the brain. All right, so it's the brain doing the work. The photoreceptors are simply allowing for hyperpolarizations or depolarizations. Right? Vision is so complex. That's why we have the on bipolar cells, the off bipolar cells, the on center ganglion cells, the off center ganglion cells. And then as we go further up, we have, we also have our cones, which allow us to see different colors. And then we have receptive fields, and we have things like contrast. Right, and in this situation, that it's the very finely tuned differences in the action potential firing rates of those ganglion cells that our brain miraculously turns into vision. But on the photoreceptor level, it's just ions coming in and out of the cell.
So basically the fact that it's like a whole cycle of inhibition and then we can get like this specific GMP just turn on and off, sends that action potential to
Yeah. So there's kind of built-in mechanisms so that, like, right, when you go into a dark room, if you didn't have the ability for those photoreceptors to kind of adjust and reset, you would just it would just be purely dark. But there's kind of a built-in system to allow your photoreceptors to sort of reset and make you a little bit more sensitive to the situation. That's what allows us to see, like, our visual world isn't just light or dark, right? If someone were to just dim the lights in here really slightly, we would all notice it, because it's kind of all about these relative levels and the structures going on that we've discussed that the brain takes and computes what we're supposed to be seeing. Yeah.
I have a question on one of the homework questions. Okay. It was number 7. It was asking, like, if we're rubbing like glass and then a brick on our skin, which skin receptors are going to be like the most activated in response to that?
Yes. What do we think? Okay, not the Pacinian. Why? Those are for deep pressure or strong vibrations. Okay, so gently rubbing your hand against the surface is not going to activate something. It's not deep pressure. It's probably not going to be a very strong vibration either, but it could be. What kind of vibration? Yes. All right, so we could have, yes. All right, so me rubbing my hand against this wall, right, I feel the texture because as my hand kind of moves over these bumps, it's kind of vibrating my skin a little bit. But there's no deep pressure here. So I would say a lot of that definitely has to do with the Meissner's corpuscles. Our Merkel's discs are also important for light pressure, so they could be involved in that as well. In light, like just gentle touch. What else we got?
Would the same ones be activated for when you touch on the brick?
Yes. Yeah.
So they're not really a big difference.
No, yeah. It would be the same. The question was really more saying, like, how do I know, like, I could close my eyes and kind of identify, okay, this might be a brick, this might be something. I can certainly tell, like, if I had two surfaces and I closed my eyes, I could certainly tell which was a brick and which was glass. And it's because of those receptors, right? So if it was glass, it's a really smooth surface. There may be a little less input from those Meissner's corpuscles because I'm not really sensing vibrations, but there is a little bit of overlap in that both of those are the ones that are kind of critically responsible for our sense of light touch or gentle touch. Yeah.
Could you differentiate between like the M-type and P-type ganglion cells that we just read in chapter 10?
Sure.
Oh, sorry, it was chapter 9.
So our M and our P-type ganglion cells have kind of two different things we can look at. One is just their actual physical characteristics is that the P-type are smaller, the M are larger. Okay, the M-type, they respond. So we see when they're activated. These are ganglion cells. So these are the ones that are going to send that information up to the brain, right? They kind of have a really quick, strong response. But as long as that stimuli is still there, in this case, we're saying the light is on, it kind of slows down its firing. All right. So they're really our M-type are kind of more useful in our immediate knowledge that like a light has been turned on or off. Our P-type are the ones that are telling us, okay, the light is still on because that stimulus is still there. And so they have longer, a longer period of time. As long as that stimulus is there, it might diminish a little bit, but they're still firing off those action potentials. Okay? And I don't need you to know, right, we talked a little bit about the visual pathway going from there. So the other consequence of having these sort of different types is organization, okay? So we talked a little bit about the LGN, right, of the thalamus. This is where ultimately that information from the ganglion cells will end up synapsing on the lateral geniculate nucleus in the thalamus, okay? And this thalamus has a very ordered structure, and part of that order is actually which cell types synapse onto which layers of this, okay? Right, it's less important to know, like, oh, P-type synapse here, because we haven't really talked about what that does. I'm just trying to illustrate the fact that all the way up to the brain level, we have these parallel pathways that sort of contain their own little bits of information, whether it's, okay, this is, this is color, this is, this is just light, this is from an on center or an off center, or we're perceiving contrast here. And then we talked a little bit about some higher order characteristics. But all of that information kind of stays segregated up to the brain, where it's really in the primary visual cortex where that information is finally turned into what we perceive as vision. We talked a little bit about, same thing here, like I'm not going to ask you to memorize, what are these layers going on? I just want you to understand that those layers maintain themselves all the way up to the striate cortex, and even a little bit from here, right? The projections that go from the visual cortex or the striate cortex from different layers are sent out to different places. Right? I just mentioned it, so I'll bring it up. We said that there's also these higher order characteristics that become present in our neurons as we travel these pathways up to the brain. Do we know exactly how this works? No, but we do know that we can, if we stimulate or sense the firing rates of individual neurons, some of them display this type of orientation selectivity, where if you had just a bar of light, depending on its orientation as it rotates, it's really sensitive to this, and it's hardly at all sensitive to that. Right? And that's just one of the millions of these cells that are in your brain helping control vision. So some are sensing, it's kind of this angle. Other ones are probably sensing other angles. And all of those little bits of information are coming together in our brain to allow us to see what's going on. Right? Allows us to distinguish things when they're, I don't know, I guess sometimes when I'm making slides or something, or you're trying to line, we can perceive the tiniest little difference of things that aren't, perfectly lined up. And if we didn't have neurons that have, some of these properties, there'd be things like that we may not be able to perceive because, we don't have neurons with those properties.
So the off and on centered ganglion cells,
yes.
Do those interpret information from both rods and cones, or just rods?
From both.
Okay. And I assume the other ganglion cell, the P-type and M-type ganglion cell, also interprets information from both rods?
Right. Yeah, the main difference between our rods and cones is that our rods are very sensitive to even low levels of light. So really, if we're in a dark room with very little light, right, we can't see colors. Part of that is because our cones are not that sensitive, and so we do pick up on every little speck of light, right? In your room at night trying to sleep, and there's just one light on something on, and it drives you crazy. We can pick up on even the little tiniest bit of light, but it's our rods that are doing that. As we move into pure daytime light, our rods are so oversaturated that they're not really contributing much information anymore. It's our cones that are, helping us perceive all the different colors of light that are giving us vision. We talked a little bit about the fovea. Let me see. Can find it. And our distribution of rods and cones as we move away from the fovea. So in the center of your visual field is where that fovea is. The retina is actually thinned out at this layer, and that allows really direct access to cones. Even though these other cells are pretty transparent and light flows through them, at the fovea there's nothing to go through. The light comes into your eye and it directly hits onto these photoreceptors. At our fovea, we have nearly all cones, okay? So that's why during daytime, our center of our visual field is able to be the most clear, right? I can see, I'm looking at, the skeleton here, I can see it really clearly, but I can't see, I can't even quite distinguish colors as well in my peripheral visual field. I might be able to say, oh, that's blue, but can I distinguish like the little gradations? Probably not, because we have less cones operating on our peripheral retina. Our peripheral retina is a big part of just sensing those little bits of light that we perceive in the dark. Okay, so one of the examples in the book is if you've ever gone stargazing, go find a spot really dark, you have to adjust a little bit because your eyes need to get used to the darkness, and you look up and wow, beautiful stars all over the sky. And they kind of say, if you look at one spot and you just pick a star in your peripheral vision and you kind of focus on it without turning your eyes actually to the star, and you focus on it for a little bit and then you do turn your eyes to the star, often that star will disappear because in the center of your visual field are almost solely cones. And at night, in the dark, your cones they're not sensitive enough to pick up on that. And so sometimes, depending on the, absolute light and darkness level, if you pick a really bright star, it probably wouldn't do that. But if you pick maybe a more faint star, you could see that phenomena. That's because in our center of our visual field, where our fovea is, we really don't have rods. Anything other than vision, or if there is vision, you can still ask. We spent a lot of time talking about vision, which is appropriate. There's 2 chapters on vision, and it's a bit of a more complex subject. Yeah.
This is actually about taste.
Okay.
With the salt-sensitive taste cells, we talked about how there's those sodium channels, and they're dependent on amiloride, I think is what it's called. Are those
correct?
So are those sodium channels specialized into where they're only found on these taste cells?
Say that again.
I remember you were saying how these sodium channels are special in a way because they're dependent. How does that work again?
You mean on this amiloride?
Yeah.
Yeah. So there's nothing special about amiloride. It's just that these channels are sensitive to it. So it gives us a way to classify them over ones that aren't. So I wouldn't worry too much about that, what the consequence of that is. It's just the fact that they are blocked by the amiloride is why we call it, that's how we can differentiate them from ones that aren't. Same thing of why we say the AMPA and NMDA receptors, right? There's, do we really care which ones are blocked by AMPA or which ones are blocked by NMDA? Not really, but it shows us that there are kind of 2 categories of them. And so this is sort of just saying that there's a special category of sodium selective channels that mediate, among probably other things, salt-sensitive taste. Yeah?
For the sour pathway, do we need to know exactly how the hydrogen ions block the potassium?
No, because we don't really even fully understand that. Just the fact that that also contributes to this pathway is that not only are we bringing our positively charged protons into the cell, but we're somehow also preventing the positively charged potassium from getting out. So both of those things mean we're more positive inside the cell, meaning depolarization. Yep. What else we got? Yeah.
Can I ask a sound related?
Sure.
Can you explain a little bit what phase blocking and locking is? I was kind of confused.
Sure. Okay. So we have this whole setup, right? Sound waves come into the ear, they turn into vibrations, they turn into fluid movement, they turn into kind of deformations of this membrane that activates hair cells, and all of a sudden we get neural signals sent from these hair cells out through the pathway to the rest of the brain. Right? That's how we sense sound. But the question is if this is the case, well, how are we sensing a really high-pitched sound from a low-pitched sound? Right? If all of it ultimately, upon, transduction in the hair cell, all of it just turns into these little neural signals. So how does our brain perceive this? There's a few things. One is that this basilar membrane makes kind of a tonotopic map, where across it, it's different hair cells, although the hair cells themselves are not special. They don't respond to it. They're just at different places on the membrane, and the membrane will deform at different places depending on the frequency. Okay, so we have that sort of set up, and this is sort of the same thing as vision, is that part of this map moves on all the way up through to the brain. And so there's this kind of idea of, okay, we know that if this line, this pathway gets activated, it ultimately came from here, meaning this sound is a 16 kilohertz sound. But there's a few other mechanisms that also help our brain figure out, well, what's the frequency of the sound that we're hearing? And one of them, especially when we're trying to distinguish low frequencies from each other, is this idea of phase locking, right? If a sound wave has these different phases, right, and it's of a characteristic frequency, what can happen is maybe we have a neuron that fires an action potential every time the sound wave is at the peak, right? So every peak, that neuron's firing another action potential. And in that way, the actual rate of action potential firings is equal to the frequency of the sound that's coming in. So that's the general idea of phase locking. Right, at higher frequencies, phase locking doesn't really help us because, even if they were to lock onto a specific part of the sound wave, the frequency of sound is actually faster than our action potentials can be fired off by the neurons. So yes, maybe you have it, still in a regular pattern, shown here, right? It's still kind of at, this is clearly not phase locked, but even if it was, there's all sorts of things going on in between here. So that doesn't do any good to our brain in trying to determine what the frequency that we're hearing is.
So phase locking has to be firing at the same point of the frequency every single time.
Correct.
Like you can't have a gap in between. Then it's not phase locking.
Right. The only caveat to that is sometimes it's a group of neurons that are sort of collectively phase locked. And so this diagram here is trying to show that in B. All right, here we have a phase locked neuron, but it's not firing at every single peak of the sound wave. It's only firing at some, but when it does fire, it's at the same spot. All right, and so this neuron might be one of a group of neurons that are all phase locked, and collectively, there's probably one firing on every single peak. So it doesn't necessarily have to be in one single neuron. It could be within a group of neurons, but that's exactly what happens. We get a firing of an action potential at the same spot in every single peak of the sound wave, or part of the sound wave. Yeah.
I just wanted to know what cell was the auditory nerve connecting to in the cochlear nucleus. Like if you go back a couple of slides, I think it's that. Yeah. Do we need to know what type of cell it is, or?
Yeah. I mean, we can look at the pathway. So the synapse here in the medulla at two spots. All right. So they first go to the medulla, then they go up through the thalamus to the brain.
But that's enough.
Yeah, I mean, I hesitate to say these things because then people are like, well, you said this and it wasn't going to happen. I hate asking you questions that are like, you know, if I were to ask you like all these different levels, right? We live in the information age. You can, if you needed to use this information, first of all, you'd probably use it so much that you just did memorize it. But if it wasn't something that you immediately you could like look it up, right? So I want you to understand more sort of just the big picture levels where things synapse versus, you know, I don't want you to spend your time remembering, okay, the superior olive and then the inferior colliculus. What would be better is to understand, okay, the spiral ganglion cells synapse in the medulla, and in the medulla, they actually synapse to both sides. So now we know that that information is going to run in parallel up to both sides of our brain, and that happens in the medulla. Right, so you could, you know, there's, right, so then if I had said something about like, okay, you receive a sound from directly into your left ear, all right, and somewhere along the way, let's say up in the auditory cortex, you have damage to your left auditory cortex where that pathway leads. Would you still be able to perceive the sound? The answer is yes, because it moves over to both sides of the brain. So that's kind of an example of, right, understanding what's going on versus just memorizing like, oh, this, that.
So it connects to the dorsal, so like we can kind of say like the back and the front.
You mean right here?
Right, and then it also connects left and right.
Correct. And remember, with this diagram, I'm just showing you one of these pathways, right? This would also go elsewhere too. But it's very complicated when we start to look at every single synapse of where this information goes. It gets too complicated for us to really even understand. So just one example is showing that, you know, it does synapse here and then that information goes to both sides up to the brain. Right, that also plays into our ability to, let's see, localize sound a little bit, right? Because we have kind of a time delay, both physically, with how the sound reaches one ear, a little bit tinier, a little bit faster than it reaches the other ear. Okay, our brain can perceive that. If we go back to the diagram we're just looking at, let's say we had just a really quiet sound that was so quiet it was in one ear and the sound can't even travel to the other ear. Well, that information is now both going to both sides of the brain. So how do we know it's coming from our left ear, or I'm using my right ear, when the physical sound wave doesn't even reach my other ear? And part of it is because even though it's minuscule, right, we're going to have a time difference between how fast the axon or the action potential can reach here and how fast it can reach here. There's a teeny time difference, and our brain can take that information and tell us, even though the sound is being processed on both sides, I know where it's coming from, right? There's other context clues to that as well, right? You know, I don't know, sometimes if it's you're really quiet, you can almost feel somebody whispering in your ear. So it's like you might have actual hair cells, not just like you know, hair cell neurons, but physical hairs in your ear that help you distinguish that the sound is coming from that ear. But our brain can make sense of those teeny tiny differences. All right, who wants to ask one last question? Anybody? What if I said whoever asks the last question gets something special?
Extra credit.
All right, if there's no questions, that's it. Passes over. I can